Hello everyone and welcome to today's live broadcast, Regulation of Microtubule plus N Dynamics during Axon Guidance and Cell Migration, presented by Dr. Laura Lowry, Assistant Professor of Biology at Boston College. I'm Susie Valdez of Lab Roots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Lab Roots. Lab Roots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Click on that green Q&A button located at the lower left of your presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of our presentation. Please also notice that you will be viewing this presentation in a slide window. If you'd like to enlarge that window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, click on the support button at the top right of your presentation window or use that Q&A button to let us know you're having a problem. Good news, this presentation is educational and offers educational continuing credits. Please click on that button in the bottom left-hand corner and follow the process to obtain your credits. Without further ado, please join me in welcoming Dr. Laura Lowry. I will now turn the presentation over to her. Welcome, Dr. Lowry.
Thank you very much, Susie. It is such a pleasure for me to have this opportunity to tell you about the research that's going on in my lab. I've been an assistant professor in biology at Boston College for about 4 years now. The big picture research question that has fascinated me for as long as I can remember is how do cells move? How do they find their way? How does a cell move towards the right direction, whatever that may be, and away from the wrong direction? Now, this is a fundamental question in cell biology. Cell movement happens in the embryo during development. It happens in the adult during wound healing, in the immune response, and it happens inappropriately during cancer metastasis. I want to make note that these movies aren't going to play in this presentation, but if you want to see any of these movies live, please feel free to email me, and I'm happy to send you clips of them so you can watch them in your own time. So cell movement is critical in development, and there can be disastrous consequences with a variety of pathological situations if cell movement happens inappropriately. Now, cell movement is particularly critical during embryonic development. Embryonic development requires both collective and individual cell movements. Many different cell types have to migrate over a complex embryonic terrain to get to where they need to be for proper tissue assembly and organ formation. In this cell movement process, there is a machine inside cells that drives the movement, and this machine is called the cytoskeleton. There are two critical components of the cytoskeleton that my lab focuses on: actin filaments, which drive cell shape and cell motility, and microtubules, which provide cell polarity and organization. Both of these cytoskeletal systems are critical for cell movement. My lab has been focused particularly on how the cytoskeletal machine is regulated during embryonic development and specifically in nervous system development. I've been studying aspects of the nervous system for about 17 years now. And within nervous system development, there are two different kinds, at least two different kinds of cell movement. First, every neuron might be born in one location, for example, the neural tube, but then have to migrate to a different location, for example, a particular layer in the brain. And then once it performs this cell migration process, then each neuron sends out an extension of itself called an axon and sends it over long embryonic distances to make the essential connections that wire up the nervous system. Now, obviously, wiring up the nervous system with accuracy and precision is vital to our normal physiological functions, and defects in this process lead to a long list of neurodevelopmental disorders, including we now think it contributes to autism and schizophrenia. And understanding how these connections form may also allow us to design more targeted strategies for promoting regeneration after injury. Now, in forming these connections, The primary player in this process is the growth cone. This is the hand-like dynamic structure at the tip of the growing axon. It's the growth cone's job to find its targets, and my lab focuses on how it moves in the right direction and how cells move in the right direction. And to answer these two questions, my lab uses the model system Xenopus laevis. Now, I thought very carefully about which model system to use for my lab. Over the last couple decades, I've extensively used C. elegans, I've used Drosophila, I've used zebrafish, I've dabbled in mouse and chick, and what I came to realize is that there may be no other vertebrate model system that has primary embryonic cells as large and as easy and inexpensive to culture, manipulate, and image as Xenopus laevis, particularly for looking at cytoskeletal dynamics. And so, since I have a large lab with many students and they all want to have their own embryos every week to do their experiments, Xenopus laevis is a fantastic model system. We can collect hundreds to thousands of eggs every week, fertilize them, inject constructs for gene manipulation, dissect out the neural tube or other cell types we're interested in, plate those cells, and then, for example, with neurons, the axons will grow out, and their large growth cones allow for clear visualization of individual microtubules and complexes that regulate microtubules. Now, when my lab began in 2014, our primary focus was on investigating microtubule regulators and how they played a role during axon guidance, and a large subset of my lab still focuses on this project. But over the last 2 years, I've been fortunate in that my lab has expanded in size and scope in terms of our funded research projects. So I'm going to spend the first half of this talk telling you about the microtubule regulator and axon guidance project, focusing on the key highlights we've made, the key insights, and then I'll talk about these new directions that my lab is going in. If you think about the growth cone, its primary function is to make decisions, whether to move forward, to retract, or to turn. The scientists have been studying axon guidance for over 100 years, but we still don't understand how the growth cone steers in the right direction. Now, that's not to say that we don't know anything. We know a lot. Classic genetics and molecular studies have already given us lots of guidance cues. We know receptors that are on the growth cone. We know signaling pathways that transduce those cues. We know a lot of the molecular players. There's also been some really beautiful descriptive biology of growth cone cytoskeletal dynamics. But what's still missing after all this time, is the linkage between the two. The emergence of an integrated picture of the cytoskeletal machine in the context of its possible effectors. How do its effectors spatially and temporally influence cytoskeletal dynamics to steer the growth cone in the right direction? I mentioned there are these two critical elements of the growth cone cytoskeleton. F-actin is involved in growth cone motility, and microtubules promotes axon outgrowth. But if the growth cone is going to steer in a directed manner, these two cytoskeletal systems have to be coordinated. And one place to look for that is where they physically interact. And so there are a group of dynamic microtubules that explore the growth cone periphery, and they can perform this internal growth cone exploration by utilizing their property of dynamic instability, where microtubules switch between growing phases and shrinking phases. And by rapidly switching between these two states, they can effectively explore the entire growth cone space and even probe into these filopodia, where guidance cues are first detected. At the tips of these microtubules are a family of proteins called +N tracking proteins, or plus tips, and they regulate microtubule dynamics. It's known that plus tip activity is directly modulated by upstream kinase signaling, and not only do plus tips regulate microtubules, but also they bind to F-actin and structures such as adhesions. And so it seems quite plausible that plus tips might take a central role in orchestrating the growth cone response to guidance cues. But the details for how this might occur are largely unknown, as plus tip interaction and function in the developing nervous system has not been extensively examined. When I was in my postdoc at Harvard Medical School, I performed dual genetic and proteomic screens in Drosophila, which is a great screening platform, to identify new microtubule regulators that were playing a role in the growth cone. I was very fortunate to have a successful screen. I've taken all these screen hits to my own lab, but I decided to start by focusing on XMAP215 and TACC3. It was known that they directly interact through their C-terminal domains. And these two proteins, they regulate microtubule dynamics at the mitotic spindle and during cell division. And it's been known for over a decade that they do this. But their potential functions within the developing nervous system had not been examined previously. Now, XMAP215 is the best-known catalytic microtubule polymerase. It binds to tubulin dimers in the cytoplasm as well as the plus end, depositing the tubulin dimers onto the microtubule to grow the microtubule. In addition to this plus end localization, XMAP215 is also located at the centrosome from where most microtubules nucleate. And it's thought that it's there so that it can load onto new microtubules and promote their growth. This is where TACC3 enters the scene. TACC3 was not described as a plus tip. It actually was published in the literature that it did not bind to microtubule plus ends. TACC stands for transforming acidic coil domain. And its function was that it was a centrosome adapter. And its main microtubule regulatory behavior was that it could bind to XMAP215 and recruit XMAP to the centrosome so that their XMAP could load onto microtubules. So TACC3's function in regulating microtubules was thought to be very indirect. But I had a sense that this was not actually the case. The studies which showed that TACC3 was not a plus tip had some experimental flaws. And so I wondered if TACC3 actually could function as a plus tip, and does it regulate microtubule plus end dynamics more directly than simply recruiting XMAP215 to centrosomes? Those were the first two questions that I decided to start with. So before I get into the function of these regulators in axon guidance, I want to talk about my lab's initial attempts at exploring the role of TACC3 in regulating microtubule dynamics. And this was spearheaded by the original Lowery Lab team, Belinda and Anna, who were postbacs at the time. Beth and Berju were my first two PhD students, and Matt, Patrick, and Aaron were undergrads. They've all gone on now to med school and grad school. So when Beth sat down at our spinning disc confocal, the first time she ever sat down at this microscope in our lab, and she was imaging GFP TACC3, and she saw that TACC3 was accumulating at the tips of these microtubules in growth cones. Here at the bottom, there's this montage of a microtubule over time. You can see that TACC3 GFP is labeling the end. And this plus end localization was not specific to neurons. Other cell types also showed the plus end localization. So in the bottom you could see these red polymers. Those are microtubules with the green puncta at the tips of them. That's TACC3. And this was the first evidence in a vertebrate system that TACC3 could function as a plus end tracking protein. Again, movies of this are you can find on my lab's website. Now we noticed that the TACC3 localization on the end of the microtubule really looked like a puncta. And this was in contrast to many known plus tips at the time, which had a comet with a long decay. And that led us to wonder, well, where exactly is TACC3 on the end of the microtubule? See, what I haven't mentioned yet is that the plus end is home to lots of different plus tips, and they don't all bind to the plus end by the same mechanism. For example, XMAP215, which I mentioned, is at the distalmost tip. It was thought that it was the only one out there, so that it's adding on new tubulin dimers. Whereas EB1 was about 500 nanometers behind XMAP215. And CLASP, which is another plus tip, overlapped with and was further behind EB1. And so we wanted to know, where is TACC3 on the plus end? We performed high-resolution co-localization analysis and saw that TACC3 was in front of EB1 and overlapped with XMAP215. And this was very exciting for us as a new lab because at the time, no other plus tip had been shown to localize at the distalmost tip with XMAP215. And it suggested that perhaps TACC3 was functioning with XMAP215 to promote microtubule polymerization. To address this question, we wanted to analyze microtubule dynamics when TACC3 was manipulated. And in my lab, the way that we assess microtubule dynamics is by using a software package which was developed in the Danuser lab where I did one of my postdocs. And this software package automatically detects, tracks, and quantifies plus tip behavior in a time series. It detects the comets as these blue dots, links them into trajectories in these red lines, and then extracts a series of quantitative parameters that quantify, describe microtubule behaviors. And using the software package, we found that knocking down TACC3 leads to a small but significant reduction in the velocity of microtubule growth, whereas overexpressing TACC3 leads to an increase in the velocity. Lifetime was not affected, so catastrophe rate of the microtubules was not affected, but the polymerization velocity was. And this leads us to the obvious question of how is TACC3 affecting microtubule polymerization? And this is the PhD thesis project of my grad student Garrett Camarada, and he has the help of some undergrads, Ben, Quinn, and Helen. And they've been asking a series of questions, including, first of all, is TACC3 recruiting XMAP215 to plus ends? I'm not going to show you the data, but the answer is yes. TACC3 is indeed contributing to XMAP215 recruitment to the plus ends, and that's one of the ways that it's modulating polymerization dynamics, but not the only way. So we also want to know, is it promoting XMAP215 polymerase activity, perhaps by binding to it that might promote this activity? Also, we're curious if TACC3 might have an XMAP215 independent role. And then finally, we want to explore whether TACC1 and TACC2 also play a role in regulating microtubule dynamics. When we started studying TACC3, very little was known about TACC1 and TACC2, and we wanted to explore those as well. So first, to get at the question of whether TACC3 is directly promoting polymerization as well as whether it's contributing to XMAP215 polymerase activity, Garrett has set up in our lab in vitro microtubule reconstitution assays of purified proteins. And what he's found thus far is that TACC3 alone in incubations with tubulin preps leads to an increase in the microtubule elongation rate. And this suggests that TACC3 might be directly promoting polymerization, but we still have some controls to run. These studies are still quite preliminary, but we're looking forward to seeing what the repeat experiments show us in the future. Additionally, I mentioned there are these other TACC members, TACC1 and TACC2. Now, when we started working on TACC3, at the time, in the literature, it was stated that Xenopus laevis only has one TACC member, and in fact was the only vertebrate model system that only has one TACC member. But I, again, had a feeling that this was not correct. When I went back and looked at the literature, the papers that said this cited previous papers that went back quite a few number of years, even before there was the Xenopus laevis genome known. And so I decided to look at the current genome, and I found indeed that TACC1 and TACC2 have been annotated in Xenopus. And so I thought these would be great undergrad projects where we would clone these genes and then look to see where they localized in the cell. And again, very little was known about the functions of TACC1 and TACC2 in any system, and so these would be really important insights to understand for the TACC field. What we found first is that TACC1 indeed can act as a plus tip, just like TACC3, and that TACC2 can act as a plus tip as well. And just like TACC3, manipulating TACC1 and TACC2 did lead to changes in microtubule dynamics in a variety of parameters. Now, we wanted to know whether there was redundancy or compensation among these TACC members, and so we started by looking at using an in situ hybridization to see where these TACC members were were expressed. What we found is that TACC3 was particularly expressed in the nervous system, in the neural tube. If you look at panel L here, there's a box around the neural tube localization, as well as these arrowheads point to the pharyngeal arches, where neural crest cells are moving down to populate the face. Whereas TACC1 and TACC2, they didn't have that neural tube and craniofacial neural crest cell localization for their expression patterns, but they were highly enriched in the somites and in other certain areas of the brain, including the mandibular nerve. So my lab is currently in the process of exploring the unique and collective functions of these TACC proteins during development and in regulating microtubule dynamics. And so now that I've spoken about the functions of these TACC family members in terms of regulating and promoting microtubule dynamics, I'm going to talk about how they're playing a role in axon guidance. This has been the project of my PhD student, Burcu Erdan, and she's had the help of two talented undergrads, Ben and Eric. And what we found is that overexpressing TACC3 and then plating these neurons and culturing them to see how they grow, overexpression of TACC3 leads to longer axons. And what Burcu found through time-lapse imaging was that the longer axons were not due to faster outgrowth velocity. And in fact, there was actually a mild reduction in outgrowth velocity. But instead, these axons, they had a clear reduction in the retraction rate. So under normal situations, control axons will spontaneously retract at a certain frequency because axons are subjected to a number of actin-myosin contractile forces within the axon itself. But with TACC3 overexpression, this retraction rate that was spontaneous was almost cut in half. And so these axons would grow more persistently over time, and so after a day of growing, the axons would end up being longer. We wanted to know if this change in persistence could reflect how axons might respond to guidance cues. When we bath apply slit, which is a traditionally repellent guidance cue, most control axons, more than 60% of them, will immediately retract in response to this repellent cue. But when TACC3 is overexpressed, only 10% of the axons will retract. And this again shows that TACC3 overexpression desensitizes the axons to repellent forces. Now, we know that slit is a soluble cue that the growth cone encounters, and somehow TACC3 is mediating the growth cone response to slit. So one question is, well, how is it doing that? It's already known that slit binds to a receptor called ROBO on growth cones, and ROBO leads to the activation of a tyrosine kinase called abelson tyrosine kinase. And ABL could potentially phosphorylate TACC3 and perhaps change its function to then mediate its response in the growth cone. We wanted to test this, and so our collaborators at University of Vermont did some kinase assays, and they found that when ABL is present with TACC3, TACC3 has a phosphorylated tyrosine residue on this right column here that is not present when TACC3 is there without abelson tyrosine kinase. So somehow the presence of ABL is leading TACC3 to be phosphorylated. We don't know yet if it's direct, but we do know that TACC3 is phosphorylated in response to ABL, and that may lead to changes in how TACC3 modulates microtubule dynamics. And so Burcu is in the process of making mutants of the tyrosine residue to see how that changes TACC3's function to regulate microtubule dynamics and growth cone behaviors. And so our model for TACC3 function during axon guidance is that in the growth cone, maybe on one side of the growth cone, you could have a repellent guidance cue, and that would lead to TACC3 phosphorylation at a certain site, and then TACC3 would not recruit XMAP215 to the plus ends, so the microtubules on that side of the growth cone would be unstable. But on the other side of the growth cone, TACC3 would recruit XMAP215 to the microtubule plus ends. The microtubules would be able to grow, and then the growth cone could turn in that direction. But in the case of overexpression of TACC3, there is so much TACC3 overloading the system that TACC3 will bind to the plus ends even when they're not supposed to, and then promote XMAP215 localization to plus ends and microtubule polymerization. And so even though repellent cues are present, TACC3 the growth cone is desensitized to that, and so you'll have axon outgrowth. And so Burju is currently in the process of testing this model in more detail. Now, at this point, I want to switch gears and talk a little bit about XMAP215 function in growth cones. I told you that it's the classic microtubule polymerase, and so you would predict that more XMAP215 function would lead to faster microtubule polymerization, and reducing XMAP215 function would lead to slower microtubule polymerization. Now, I actually studied XMAP215 a little bit during my postdoc, but when I knocked it down, the plan had been to knock it down and see a slower polymerization and then look to see how XMAP215 was spatially regulated in the growth cone. But during my postdoc work, when I knocked XMAP215 down, I did not find that microtubule plus ends were slower. And actually, the microtubules themselves were moving in some patterns that were not expected. And because it was a little bit confusing at the time, I decided to put that project on the back burner as I started my lab and focus on TACC3, which had more of an expected phenotype when I knocked it down. But then I knew I wanted to eventually get back to XMAP215 because I could tell that something was really interesting about XMAP215 in growth cones, that it might have a novel function that had not been previously explored. And so a year ago, I had a postdoc join my lab, Paula Slater, and she had two undergrads working with her, Annika and Ali, and they decided to explore XMAP215 function in growth cones. And the first thing that Paula noticed when she knocked down XMAP215 and stained it for just actin and microtubules, she saw that the XMAP215 growth cones were larger than control growth cones. You could see that the entire growth cone area was larger, as well as these philopodia, shown in green, were longer as well. And these two phenotypes could be rescued by adding back XMAP215 mRNA, showing that these phenotypes were specific to XMAP215 knockdown. Now, this green, this is actin. And so what this data suggests is that XMAP215 is somehow, perhaps indirectly, affecting F-actin. But the question is how? Now, here is a movie showing XMAP215 GFP, and you can see that it's bound to the plus ends, it's enriched at the plus ends, and it's also along the microtubule lattice. But it's not obviously enriched in the F-actin networks in the periphery of the growth cone. However, these microtubules that you see are actually growing along F-actin bundles. And in order for the microtubules to grow along the bundles, they have to be coupled in some way to them. However, F-actin undergoes retrograde flow continuously. And if the flow is coupled to microtubules completely, the microtubules will loop and will not be able to go into the periphery. And so we wondered if XMAP215 might somehow be regulating frequent transient couplings from the F-actin. So what Paula did is she looked at microtubules and she used super resolution microscopy to carefully quantify the behavior in the localization of these microtubules within the growth cone periphery. And what she found is that in control situations, almost all microtubules are growing along F-actin bundles. But in the XMAP215 knockdown condition, a significant proportion of microtubules shown are going off track. You can see there with the arrowheads here that those microtubules are not along F-actin bundles. And again, this could be rescued by adding back XMAP215 mRNA. And so not only are there more microtubules that are off-roading, that are not on the F-actin tracks, but the penetration of these microtubules into the peripheral domain of the growth cone is also increased with XMAP215 knockdown. And so right now, Paula is in the process of deciphering the mechanistic function by which XMAP215 can couple microtubules and F-actin, and we're very excited to see what this work shows us in the future. So now I'm going to focus on these newer projects in my lab on cell migration and other disorders. Neural crest cells are a multipotent migratory embryonic cell type that they first derive from the neural tube, and then they undergo an epithelial to mesenchymal transition to then migrate in very characteristic patterns to become craniofacial structures, heart tissue, skeletal tissue, different muscles, and peripheral neurons. And the work in my lab that has been focused on neural crest cells has been spearheaded by my PhD student, Beth Bierce. Now, because these neural crest cells undergo an epithelial to mesenchymal transition, and metastatic cancer cells also are thought to undergo a epithelial to mesenchymal transition, I was able to convince the American Cancer Society to fund my lab's work in investigating microtubule regulators and how they might be playing a role in neural crest cell migration as a model for general cell motility. Now, we became interested in neural crest cells when Beth was sitting at the confocal imaging GFP TACC3. Again, she was the person that first saw this +M localization. You could see the GFP TACC3 is located on these, where the arrowhead's pointing, at the ends of these microtubules. But she also saw that in certain populations of cells, such as neural crest cells, GFP TACC3 was highly enriched at the leading edge surrounding focal adhesion structures. Now, we since have come to the conclusion that we think that this localization might be partially an artifact due to exogenous expression. However, it led us in the direction of looking at neural crest cells and how they might be playing, how TACC3 might be playing a role in their migration. So the first question we want to know is, well, is TACC3 expressed in neural crest cells? And when we did an in situ hybridization for TACC3, we found that indeed, TACC3 was highly expressed in these craniofacial neural crest cells. These arrowheads are pointing to these three arches. Those are the pharyngeal arches on the side of the face, and you can see in the dark purple that the TACC3 is highly enriched in this location. And what we found is when we knocked down TACC3, we saw it and then used a different marker for craniofacial neural crest cells, such as SOX9. We found that the morphology of these pharyngeal arches was quite disrupted, including there was a reduction in the length of these arches. And then when my student Beth took these cells and cultured them and analyzed the parameters of cell motility, here on the left you could see an explant that she cultured, and then she did time-lapse imaging to look at the motility dynamics. She found that TACC3 knockdown led to slower motility, and TACC3 overexpression led to faster cells. And now she's in the process of examining focal adhesion dynamics after TACC3 manipulation to see if TACC3 affects either assembly or disassembly of these focal adhesions, and that would be one way that TACC3 contributes to cell migration. Now, in addition to using neural crest cells as a general model for investigating cell migration, we are also looking at a particular disorder called Wolf-Hirschhorn syndrome. And this work has been spearheaded entirely by an undergrad with Ali Mills, with a big undergrad team in my lab, and they've also had the support of a senior scientist in my lab, Sang-Mook Lee. And what they've been doing is investigating the genes associated with Wolf-Hirschhorn syndrome. So we became interested in this syndrome when we learned that TACC3 is one of 5 genes that are missing, one copy, in this genetic disorder. The Wolf-Hirschhorn syndrome field has spent over a decade figuring out which genes are associated with this particular disorder, and the disorder is characterized by various craniofacial defects, including a wider face, heart defects, skeletal defects, some other issues with cognitive functioning. But what's interesting is that very little is known about the embryonic function of these 5 genes. FGFR3 they know somewhat about, but the other genes, their possible functions during development had not been characterized. And so we thought that our system would be a really great model system to finally investigate how these genes, how the proteins encoded by these genes might be playing a role during Wolf-Hirschhorn syndrome onset. And we wondered specifically if Wolf-Hirschhorn syndrome might be a neural crest cell migration disorder, or if at least that's part of the contributing factors to the phenotypes. And so first we wanted to know, well, are these genes expressed in neural crest cells? On the top left is a marker for X-twist. This is a neural crest cell marker. You can see it labels these pharyngeal arches as well as around the eye. And what we found is that most of the genes that are associated with Wolf-Hirschhorn syndrome, WHSC1, WHSC2, LEDM1, and TACC3 I've already mentioned, are indeed expressed in neural crest cells. So they're expressed in the right place at the right time to be playing a role in neural crest cell migration. So now what we've been doing is knocking down these genes individually and collectively and looking at possible phenotypes during development. For example, when we knock down WHSC1 only on the left side of the embryo, on one side, what we found, and then we did an in situ for X-twist, again this is a neural crest cell marker, what we found is that the craniofacial neural crest cells do not migrate as far down as in the control side, the control injected side. And one of the wonderful benefits of using Xenopus laevis is you can actually knock down just one side of the embryo, so you have this internal control for normal migration patterns. And so this is very obvious that knocking down WHSC1 leads to a pharyngeal arch defect, but knocking down WHSC2 does not. So this shows that WHSC1 may play a larger role in the craniofacial defects that these patients show compared to WHSC2. We also find that when we knock down WHSC1 in the entire embryo, the entire tadpole, we get a wider face, which is reminiscent of the human phenotype. And so we're excited for this preliminary investigations. Ali is in the process of knocking down all of the genes and then again collectively knocking them down to see which genes correspond to which phenotypes that occur in Wolf-Hirschhorn syndrome. So now I'm going to spend the last few minutes of my talk talking about intellectual disability disorders and what we've been doing with them. And this is been part of a really great collaboration with Dr. Santosh Girarajan at Penn State. And I gave a seminar at Penn State a couple years ago. And Santosh is a human geneticist, and he identifies genes that are associated with various human intellectual disability disorders. But one of the issues that these human geneticists have is once they identify all these potential genes, they don't know which ones are actually contributing to the brain development issues. And so as we were talking, we realized that my model system would be a very easy and inexpensive and efficient way to test the genes that he's identified. So for example, so this has been spearheaded by my newest PhD student, Michaela, in my lab. She has a big undergrad team, and again, she's supported by my senior scientist. Now, Santosh has identified a number of different deletions, including this one microdeletion on chromosome 16, and encompasses several different genes. What Michaela's been doing is knocking down these genes individually and then in combination to see what phenotypes arise during embryonic development. We want to know which genes are responsible for the various patient symptoms. What we found so far is that C16 or 52 promotes axon outgrowth. So on the top left, you can see knocking it down leads to shorter axons, and this could be rescued by adding back C16 or 52 mRNA. But knocking down the other genes has no effect on axon outgrowth. So C16 or 52, nothing is known about this gene, and so this is the first known function that is playing some sort of role in axon outgrowth. But Michaela is not just looking at axon outgrowth, but looking at other parameters as well. For example, she's looking at craniofacial defects. She has found that knocking down POLR3E, UQ, CRC2, and 16 or 52 also shows craniofacial defects, where CDR2 shows no effects at all. And then finally, these patients show microcephaly defects. And so here is a quantification of the facial width, for example. And so these patients have microcephaly defects, and so we're also looking at microcephaly in the Xenopus embryos and in the tadpoles. And here what we can find is that POLR3E knockdown on one side of the embryo only leads to the significantly smaller forebrain area. And we are excited to see continue using our strategies to investigate intellectual disability disorders. Now, before I conclude, I just want to mention that other members of my lab are focusing on other projects. Quinn and Jack are setting up CRISPR-Cas9. In the Xenopus field, people are starting to use this quite successfully, just like in many other systems, and so we'll be using this as a complementary approach to our current knockdown strategies. My student Kyle Lawrence has a great collaboration with the CollegeLaura Lab at Yale studying Abelson tyrosine kinase 2 function in growth cones. And then finally, my undergrad Jingzong Yan is looking for new embryonic functions for a previously known microtubule regulator called CyPin. With that, I want to make a number of conclusions. First of all, I want to reiterate that Xenopus laevis is a fantastic embryonic cell migration model, that TACC3 is a plus tip that promotes microtubule polymerization during axon outgrowth and guidance, and that all TACC members are plus tips that affect microtubule dynamics. Finally, XMAP215 can couple growth cone microtubules with F-actin. TACC3 promotes neural crest cell migration. TACC3 may contribute to Wolf-Hirschhorn syndrome along with other Wolf-Hirschhorn syndrome candidate genes, and my lab is now using Xenopus to investigate various intellectual disability disorders. And going forward in the future, my lab will continue exploring plus tip function during axon guidance and different types of cell migration in both normal and pathological conditions. With that, I want to acknowledge the members of my lab. I have a really large group, but we are organized in teams, and it's been very productive so far. I've had great alumni that are now on to grad school and med school and other places. I've been fortunate with having a great team of collaborators, and I've also been fortunate with acquiring funding. Thank you for your attention, and I'm happy to answer any questions you might have.
Thank you so much, Dr. Lowery, and thank you for that informative presentation and your dynamic research team. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of your presentation window, type your questions into that box that appear on the screen, and click that send button. We'll answer as many questions as we have time for. So let's get started with our audience members. Our first question today is, you are focusing on the functions of TACC3 in this presentation. Are there other TACC family members that function similarly?
Yeah, that's a great question. I briefly mentioned TACC1 and TACC2, but I didn't have a chance to talk about their particular functions. So it's really interesting. As I mentioned, they both function as plus tips, but they function in different ways. So TACC1 is actually slightly behind TACC3 on the plus end. And why it's slightly behind it and what it's doing there is still unclear. TACC2, on the other hand, is actually at the tip with TACC3. And so we're trying to understand, what these different functions that they mediate could be. So TACC1 is, as I said, it's slightly behind TACC3, and yet when you knock it down, it has the same function of TACC3. So knocking TACC1 down leads to slower microtubule polymerization. When you knock TACC1 and TACC3 down together, there suddenly is a microtubule catastrophe phenotype. So it's clear that there are both unique and redundant functions of these TACC members. TACC2, on the other hand, when we knock it down, we do not see slower microtubule growth. In fact, in some cases, we see faster microtubules. And so we still need to dissect which parts of the proteins are functioning in various capacities and regulating microtubule dynamics. But we hope to make some new insights in the future. There's a lot known about TACC3. There's a handful of papers about TACC1, and there's almost nothing known about TACC2. And so we look forward to making these new insights as we go forward.
Thank you. We have audience members tuning in from several countries today. Here's our next question coming in. Does XMAP215 also play a role in axon guidance?
That's a great question. So I talked about its role perhaps coupling microtubules and F-actin, but I didn't talk at all about what it's doing in terms of guiding the growth cone. So what I found is that knocking down XMAP215 also leads to shorter axons, just like knocking down TACC3. But as I said, we don't see the same microtubule effects. What I haven't done yet, my lab has not challenged the growth cone with guidance cues to then see if XMAP mediates the response to those cues. I would predict that it does, especially if it's involved in coupling microtubules with F-actin, because that coupling is going to be really important, as I mentioned, for whether the growth cone moves in one direction versus another. But we still have to do those assays. We have to do similar assays to what I showed, where we apply various cues and see if XMAP215 manipulation changes the response. We also need to challenge the growth cone with some asymmetric cues and see if growth cone responses are different when XMAP is manipulated. And so we are actively working on those. We have an R01 to fund that work, and we hope to find out in the near future if XMAP215 is indeed mediating axon guidance. I would predict that it is, but we have to test that and see.
Thank you, Dr. Lowery. We have time for one more question from our audience. Can you talk a little more about the Wolf-Hirschhorn syndrome and the role that TACC3 might play in that disorder?
Yeah, definitely. So Wolf-Hirschhorn syndrome has been characterized for over a decade. And again, the geneticists have been very carefully focused on what specific genes of these 5 genes are needed to be missing. But again, they're only missing one copy to lead to the various human phenotypes. And when I read about these phenotypes, there's craniofacial defects and heart defects and skeletal defects. Neural crest cells just rang a bell. And so I recently published a review with one of my students proposing the idea that Wolf-Hirschhorn syndrome might be a neural crest cell migration disorder. And in terms of what TACC3 could be doing within the onset of this disorder, since TACC3 is highly expressed in these neural crest cells, it's quite possible that knocking down TACC3 leads to changes in neural crest cell migration that then result in these various defects. However, I have to point out that TACC3 is also required for cell division, and so we need to do some more careful studies to see if cell migration or cell proliferation are affected. And so we look forward to doing that in the future. I have a March of Dimes grant as well as a National Institute of Dental and Craniofacial Research grant to fund this work. And both of these grants were, one of the grants was a pilot grant, so we're still just getting our feet wet in terms of dissecting the functions of these genes in this disorder, but I anticipate we'll be making some really important insights as we move forward.
Thank you, Dr. Lowery. Did you have any closing remarks you wanted to provide the audience members before we close?
No, it's been a pleasure presenting my work to all of you, and I encourage you to check out my lab's website. If you just search Lowery Lab at Boston College, I have all of the publications of my lab over the last 4 years and links to all of them and to the movies as well, since I know many of the movies didn't play on this presentation. And I encourage you to contact me if you have any further questions. I'm happy to help you out.
Dr. Lowery, thank you so much for your presentation and also the important work that the Lowery research team is doing in that lab. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye-bye.